Welcome to our webinar Wednesday series. I'm Dr. Keith Bramwell with Jamesway Incubator over the consulting Hatchery Consultancy Group. We started this thing several years ago and been received very good response from people, and we're glad that you all are here and participating with us. We will have an opportunity. This is translated into Spanish. We have an opportunity for you to listen to the presentation in Spanish. So I'll have Mauricio come on and explain that to those that want to hear it in Spanish, and then we'll get started. Mauricio?
Oh, of course, sir. Bueno, bienvenidos nuevamente a todos a este webinar de los miércoles. Como siempre, es un placer tenerlos por acá. A las personas que están escuchando a hispanohablantes o personas que hablan en español, que todavía estén de pronto en este canal, les recordamos que en la parte de abajo van a ver un pequeño mundo que dice Interpretation. En ese pequeño mundo pueden poner el idioma en español para recibir una traducción simultánea. Como siempre, recuerden que las preguntas las estaremos respondiendo al final y las pueden agregar en la pestaña de Q y A. Bienvenidos. There you go, sir.
All right, thank you. I don't see the little box down there, the interpretation box, but anyway, so today we've got a speaker from Cobb Vantris, Kylie Bruce Sizemore, I guess. We'll use your full name right here. She works for Cobb. She grew up working in the industry. She's got a lot of practical experience. Her family had chicken houses, and so she has had that experience. She went to the University of Georgia, got her bachelor's and master's degree at the University of Georgia. Has been with Cobb now, working in the operations of the technical services team, and she's currently also working on her PhD while she works a full-time job. So good luck with that. She'll be working on her PhD at Auburn University. So good luck and good luck with that. I'll put it that way. So her presentation today is going to be, she's going to be talking about the essentials of proper hatchery sanitation. And so look forward to hearing this. Also, one more thing, if you have questions during the presentation, down at the bottom, there's a Q&A box. Type in your questions there. We will address the questions at the end of her presentation. I'll be asking her, and we'll have a discussion at the end to get some of the questions that you have. Also, one of the questions we always get is people want a copy of this. You will get an email that'll have a link to this recording that you can watch it again later, which a lot of people tend to do, and also have an opportunity to get a PDF of her presentation if you would like. So you will all get that at the end. So with that, I will turn the time over to Kylie and let her roll here, and see you when you're all finished up. Thanks.
Thank you, Dr. Bramwell. I have shared my screen, so can everybody see my screen here? Full screen.
Yeah, looks good.
Perfect. It's an honor to be here with you all today virtually. I know this is kind of a unique scenario. I wish that we could be together in person, but today we're going to talk a little bit about the essentials of proper hatchery sanitation. I've gone ahead and split the presentation up into 3 parts today. First, we'll discuss motivation. Why should I care about sanitation? Why is it important in my facility? Then we'll move into talking about methods of achieving a proper clean and methods of proper sanitation. And we'll finish up with monitoring. What can I do to test the level of cleanliness within my hatchery? So we'll go ahead and get started first with motivation. Our poultry industry is set up in such a unique way because there are so many breeder farms spread out within our complex or our company or our integrator. They're spread out across a vast geographic region, and I think this is a great picture of that. Each of these breeder farms is owned and managed and maybe worked on by different individuals who have different standards of what the day-to-day activities should look like on the breeder farm. And all of these eggs are picked up and brought into our hatcheries. And I've heard Ben Green say multiple times that the hatchery is our little box. We can control everything that happens with our little box, but we might not be able to control what happens before we get those eggs into our hatcheries. But nonetheless, it's our job to take the eggs that we're given and produce good, viable chicks that will then be sent out to broiler farms, again, that are spread out across a vast geographic region owned and managed by different individuals, and the performance of the chicks, particularly early mortality and chick quality, is going to directly reflect our hatchery. The good thing for us, though, is that we can capitalize on each individual area of our hatchery to increase sanitation and do a great job producing a good viable chick for all of those broiler farms. Most hatcheries are set up in a unique way where the eggs are going to come in on the egg room dock on one end of the hatchery. They're going to enter into our egg room, and as we move through the hatchery, our eggs are going to flow in one direction. They're going to go into the incubators, they're going to be transferred into the hatchers, and then on the other end of our hatcheries, we have our chick pool room, our chick processing room, and then maybe a chick holding room, depending on how your facility is built. And then the chick loading dock is going to be on the other end of the building from the egg room. And this can help us capitalize on sanitation as well. As we talk about sanitation, the first thing that we have to remember is that the health of newly hatched chicks can be severely impacted if they hatch within a bacteria-filled environment. And I have 3 examples of this today. The first one is ontholitis, or yolk sac infection. Everyone here knows how the chicks hatch, right? In those last few days, the chicks are drawing that yolk sac into the navel. And as they pip and hatch, if those navels are still open, even just a little bit, anything present in the hatcher can enter into the navel and cause infection, any bacteria or fungus or mold. And this can lead to early mortality on the farm and a poor reflection of the performance of our hatcheries. The second example is aspergillosis, or brooder pneumonia, and that's caused by the mold Aspergillus fumigatus, which is pictured here during a breakout. This leads to pearly white lesions in the air sacs and primarily respiratory issues in young birds that, again, can lead to early mortality and poor chick quality that will not reflect too well on the hatchery. And finally, we have the prevalence of Pseudomonas aeruginosa, which is found in standing water, humid environments, and non-sterile vaccine lines, just to name a few examples. Just because of the general day-to-day work of the hatcheries, we can't really control the fact that we are going to have humid environments in some areas of our hatcheries, right? The tray wash room, box wash, incubators, hatchers. So we have to do everything that we can to sanitize these areas properly. So before we talk about how to properly sanitize every single area of the hatchery, we first have to understand and narrow down the sources of contamination within each of these areas that we discussed earlier in our little diagram here, because the sources of contamination are going to be different depending on where we're at in the hatchery. So we'll start in the egg room. Our hatchery will only ever be as clean as the dirtiest egg in our egg room. That's a pretty monumental statement to make, but it's so true, because if we have farm A, breeder farm A, who's sending in a great egg pack, clean eggs, no cracked eggs, their eggs look great. They're coming into the egg room and they might sit on the floor right beside a farm rack of eggs from farm B, who might send in a few floor eggs every now and then, like these pictures here, or eggs that were laid in dirty nests. And all of those dirty eggs from farm B are going to be in the same airspace as those clean eggs from farm A. So a question we can ask ourselves, are we truly monitoring the cleanliness of the eggs coming into our hatcheries on a regular basis? Or are we just going back to our egg rooms and we say, you know, farm B always has dirtier eggs than farm A? Something to consider every day that we're looking at contamination in our egg room. Another source of contamination in the egg room are the little messes that happen every single day with the setting of thousands and thousands of eggs. Obviously, we're going to have broken egg yolks. We're going to have some cracked eggs. We might drop a few on the floor. And so because of that, we're going to have a bucket or a trash can or some method of cleaning up the messes in our hatchery egg room. Now, I love these two photos because this is such a great example of how thorough daily cleaning can make a difference even in the egg room. So if you see the bucket on the left here is used on a daily basis to pick up eggs that fall on the floor during sets. Now, the folks in this egg room take this bucket at the end of the day, every single day, and they dump it out, they wash it, they sanitize it, and they take it back to the egg room for the next day. So it's ready to go the next morning. The bucket on the right obviously is used the same way to collect eggs that fall onto the ground. But this hatchery does not take this bucket up at the end of every single day, dump out the contents of the bucket, and clean and sanitize the bucket. And so it can cause buildup on a daily basis. And this can lead to poor sanitation in the egg room because we'll attract flies, and this is a perfect place for microbes to grow and thrive. Another source of contamination in the egg room can be our egg truck drivers. So obviously the egg truck drivers are an integral part of our business. Without them, we wouldn't receive eggs into the egg rooms. But when we think about the fact that they've just been to multiple breeder farms, again, owned by multiple different people, managed in multiple different ways, maybe one farmer hasn't cleaned his egg cooler in a while, but another farmer cleans their egg cooler on a regular basis. This can affect the sanitation in our egg room. So limiting the areas within our hatchery that egg truck drivers can access is vital to the success of sanitation. As we move throughout the hatchery, the next thing we come to in the process is our incubators and transfer. And this is probably the most obvious form of contamination with the rotten and exploding eggs that we have during incubation and then during the transfer process. Of course, also some eggs get cracked during incubation and they can get cooked onto the floor of the setters or the walls or the doors, and this can lead to contamination. Moving into the hatchers, probably the greatest source of contamination in the hatchers comes from the pipping and hatching of thousands of chicks within a machine. And so obviously you can see photographed here, not only do we have little particles of eggshell getting everywhere on the floors and on the baskets, but we also have a great amount of chick fluff, chick dander, that can be a great home for microbes to grow and live and thrive, leading to contamination during the hatch process. Next in line would be our chick pool room and processing room. And this contamination is going to be similar to what we see in the hatchers. It might look a little bit different, but the chick fluff that's going to roll around in our chick room floors and the particles of eggshells and chick dander on our pool belts are going to be a great place for microbes to grow. We can't talk about contamination without talking about random other ways that our hatcheries can become contaminated. So we'll start with the picture on the left here. Obviously, we want to keep a lookout for bird feathers and bird nests on a regular basis, as that can be a source of disease contamination within our facilities that can have devastating effects on our industry. Moving to the middle photo, we want to have a great hold on our bait station routines, as rodents can also carry contamination in and around our facilities. And then our third photo here is a great example of how door discipline is very important within our hatcheries. It might get hot and steamy in our box washroom or our tray washroom, and without proper education of our hatchery employees, they're just hot. They just want to cool off the room. So they'll prop that door open, but this can lead to the entrance of rodents or birds or people that shouldn't be coming in to our hatcheries. So we've talked about sources of contamination and our motivation for why sanitation is important. So now we'll move into talking about methods of hatchery sanitation. The first thing that I want to show you as far as methods of sanitation is the center circle. This 4 factors of an effective clean was developed by Dr. Sinner in 1960 in relation to how washing machines work, but it can definitely be applied to cleaning any area of our hatcheries. So there are 4 factors of an effective clean based on this model. The first is mechanical force. So when it comes to the hatchery, that can be brushing organic matter, scrubbing organic matter away, or using a high pressure water hose. The second factor is time. So in the hatchery, exposure time of a foaming chemical agent to a soiled area that we're cleaning. Number 3 is temperature. There is a great requirement for hot water for optimal detergent performance. So that would be a great example here. And then number 4 is chemistry. Are we using the proper type of chemical and the proper concentration in the area that we're cleaning? We're going to get our best results when we're cleaning if all 4 factors are optimized. And I like to use the example here of a 4-legged stool. So if I walk into a room and I see a 4-legged stool sitting in the corner and it has all 4 legs intact, attached to the stool, like the stool was built, I'm probably going to sit down on the stool without hesitating. It's the same point when we're cleaning our hatcheries. If we have all 4 factors, our cleaning is going to be optimized. And I have a few examples here. So for mechanical force, are we properly scrubbing our hatchers before we're applying sanitizer? If not, we're just going to spread all that dirt and organic material around throughout our hatcher with water. For temperature, is our tray wash actually at 130 to 140 degrees? Or our high pressure water hose that we're using to clean our hatchers? We think it's at 140, but have we gone back and looked and checked the temperature of our water? For time, our sanitizing foam hang time, we want to have the correct tip on our foaming system so that we're getting the correct consistency of foam so that we don't just spray the foam on and then in 1 minute it's already completely washed away. We want to follow the label on our chemical sanitizers. And of course, we don't want the foam to be too liquidy, but we also never want to let our foam dry on the surface as well. And then number 4 would be using the proper chemical, depending on where we're cleaning. So let's think back to our 4-legged stool model. If we walk into a room and our 4-legged stool only has 3 legs, we might hesitate to sit down, but the stool is still probably going to hold us up. It's the same thing in the hatcheries. If we take away temperature, if our hot water's not working today, we're not looking at our tray wash temperature, we don't even care. We just want to run our trays and get done for the day. Then we're going to have to increase our chemistry, our time, and our mechanical force in order to compensate for the loss in temperature. This means that if we're cleaning a hatcher and we take away our hot water, we're probably going to have to scrub harder, and it's going to be a more difficult job for us. Now let's take out one more. Our 4-legged stool now only has 2 legs, and so we're probably not going to sit down on the stool because it's not going to hold us up. The same thing applies in our hatcheries. If we take away our hot water and we take away our mechanical force, we're not going to be able to achieve an effective clean within our facilities. When it comes to different types of chemicals and their properties, the Cobb Hatchery Management Guide has a great chart here that tells the different types of chemicals and their properties. We won't get into this today, but this is a great useful tool when looking at the corrosiveness or the toxicity of different products that we use and where these products would be best used within the hatcheries. No matter what area of the hatcher you're cleaning, a basic cleaning and disinfecting procedure should be implemented. And this is just an example of what a C&D procedure might look like in your facility. The first step is always going to be to remove organic matter. It's the most important. It should take the most time out of any of the steps because it is that important. So I like to use this umbrella illustration here because when we're out on the beach and we're in the shade of our umbrella, we're not going to get sunburnt, right? Because the umbrella is protecting us from the sun. The same applies for hatchery microbes. If they're hiding behind organic matter, fecal matter, or rotten eggshells in incubators, or rotten egg exploding on the wall, then when we go in with our sanitizers, that organic matter is just going to shield those microbes, and our sanitizer will not be able to come in contact and properly kill those microbes. So, number one, remove organic matter. Very important. Number two, apply soap, scrub, and rinse. A general example of a soap here would be an alkaline cleaner, but whatever works for your facility here. Number three is apply a sanitizing foam. Again, there are many methods of applying foam, but whatever works for you would be part of your cleaning and disinfecting procedure. And then steps 4 and 5 is to rinse and properly squeegee out the area again, so we can reduce the prevalence of standing water and humidity throughout our hatchery facilities. When we think about general sanitation with disease prevention in mind, we have to remember that personnel can cross-contaminate with their clothing and their shoes and their jewelry. There are things that we can do to mitigate this, such as this automated boot spray, but it's always good to encourage and educate our employees on the importance of limiting their contact with avian species outside of the hatchery so that we can do our part to prevent disease spread throughout our facilities and our industries. Rodent control schedules are very important, as rodents can be vectors for diseases in our hatchery as well. We mentioned earlier, you can employ a bait rotation so that you're not developing resistance against certain bait products. And then if you find a bird nest, let's remove it on Friday afternoon before we go home for the weekend so that we have some time away from the hatchery, instead of removing it Monday morning or Monday afternoon when we first find the bird nest and we'll be there in the hatchery processing chicks all week. General sanitation guidelines. Number one, complete a top to bottom clean every single time. This applies to our hatcheries, but a great applicable example of this would be after we're cleaning our kitchen, after we cooked a big meal. You would never sweep your floor before you clean off your counters, because the crumbs and scraps of food that might have fallen on the counters during cooking are going to fall onto the floor when you clean the counters, and you're going to have to sweep the floor again. It's the same thing in the hatchery. We want to start at the top and work our way to the bottom for optimal clean. Because microorganisms need a nutrient source to grow, we always want to work to limit the nutrient sources available for those microbes. Number one, we can minimize clutter by having essential equipment only in the room. A good example of this, if we're having an issue with a hatcher and we send a maintenance personnel to work on the hatcher and they bring their maintenance cart with them with their tools and their toolboxes, that's fine, right? It helps save them a trip back to the maintenance shop. But what if it's time to go on break? What if it's time to go to lunch? A simple thing that we can do is roll that tool cart back to the maintenance shop so that any germs or microbes on those tools in the maintenance shop, which we might not clean as often as we clean our hatchers or our setters, won't be sitting in the room in the airspace of the chicks and in the airspace of the hatchers. We can remove cardboard packaging and store chemicals on shelves and in cabinets just to try to limit the space that microbes can find and grow and thrive in. And then, of course, we've mentioned this a couple times, but keeping areas as dry as possible, always making sure we squeegee water out of the floor after we clean. Moving on to talking about our box wash or our tray wash, it's important to think about our process for how we're drying our boxes, because we know that wet boxes will grow mold every single time if we stack them on top of each other while they're wet. So are we stacking our boxes up and giving them plenty of time to dry under the fans before we're down stacking them? Or do we have a box dryer in our facilities just to try to limit mold growing on our freshly cleaned boxes. More on our box wash or our tray wash. How often are we checking proper sanitation levels? There are several different methods of doing this. Of course, we could get the little strips, pH strips, quat strips. They can be a little bit more subjective because you just compare the strip to the color on the pack, right? But we could also buy a meter that tells us the pH. But how often are we checking our sanitation levels? How often are we checking our water temperature? We talked about that a little bit earlier. If we know that the optimal temperature of our tray wash is 140, are we writing down what temperature our tray wash is at when we start running at the beginning of the day and when we come back from lunch? We know that the tray wash is the limiting factor for how fast we can pull chicks, but it's important to have that set at a speed where we're getting a proper clean on our trays at the end of the day. An alternative to traditional methods of cleaning and sanitizing is the PathoSans machine. This machine uses H2O and NaCl to produce both a cleaner and a sanitizer. It is safe for employee contact. It can be less expensive than traditional methods of cleaning and sanitizing, but the upkeep of the PathoSans is vital to ensure the soap and sanitizers are present at the beginning of the day when it's time to turn on the tray wash or the box wash and run the tray wash. We can't have a talk about hatchery sanitation without mentioning the use of formaldehyde and hydrogen peroxide based on your facility. We could talk about this all day in a whole separate presentation, but I would just urge you to double check your formaldehyde schedule or your hydrogen peroxide schedule or whatever you use in your facility. Is your formaldehyde drop schedule still working for you? How's your kill doing in your machines of microbes while chicks are hatching? There have been so many papers published about the toxicity of formaldehyde and the corrosiveness of hydrogen peroxide, and lots of papers about looking at the use of ozone or probiotics as an alternative. But again, I would just urge you to double check on your formaldehyde schedules and see how they're working for you. We all have AON units on our hatcheries. They do breathe life into our hatcheries, and so we need to treat them as the important pieces of machinery that they are. Are we replacing our filters and cleaning our coils as needed based on the location of our hatchery? There are several things to consider here. Is my hatchery near a feed mill or farmland that has hay production a few times a year or row crop production a few times a year? Is my hatchery near construction sites? All of these will produce larger amounts of dust than if your hatchery was not near any of these. And so you might have to change out your filters and clean your AONs more often than the hatchery down the road that isn't near a few hay pastures or row crop acres. And so this is very much one of those things that you might have to just figure out what works best for your hatchery. Maybe we clean and replace our filters once a month, but down the road they replace theirs every 2 weeks. This will vary from hatchery to hatchery. Another thing to think about when we think about our AON units and how often we're changing our filters is, where are the AON units located? If they're over the pool room, we might have to change those filters more often than the AON units above our egg room, just based on the day-to-day activities that happen in that area of our hatcheries. So we've talked about motivation for sanitation, we've talked about methods, and so now we'll finish up with monitoring. I've heard Ben Green, along with his saying that our hatchery is a box that we can control, he says that if you didn't document it, you didn't do it. So what are we documenting within our hatcheries? I have a few examples here. We've mentioned this. Are we documenting water temperature and disinfectant and cleaning levels on our tray wash machine? Are we documenting and signing off on our Embrex PM sanitation check? That yes, we did run the PM cycle and we did run alcohol through our vaccine lines, again, to prevent the prevalence of Pseudomonas within our facilities? Are we checking our formaldehyde and hydrogen peroxide systems on a regular basis? Did I run formaldehyde yesterday? Yes, I sign off on my document that I did run formaldehyde and refill my reservoirs. And finally, another example is the condition of eggs in our egg rooms. So at the very beginning of the presentation, I mentioned, are we truly monitoring the cleanliness of our eggs? Are we just going and making mental notes, or do we have a sheet that our egg room attendants can fill out on a weekly basis that marks the number of dirty eggs or the number of cull eggs coming in from specific flocks? All things that we can document to prove that we're keeping an accurate check on our sanitation within our hatcheries. Of course, one method of monitoring sanitation status is traditional microbial plating. It's very useful in the hatchery because it allows us to isolate and type bacteria and fungus using different growing mediums. I love traditional microbial plating because the possibilities are truly endless as to what we can look at within our hatcheries. So obviously, there are many different methods of traditional microbial plate use, but I've listed 3 here today. The first one is surface swabs. So, after we clean our incubators or hatchers, we can go behind once they're dry and we can swab the walls or the doors, and we can streak those onto a Petri dish and incubate those in-house for 24 hours, and we can see the next day our results and how well we cleaned. We could also use surface swabs to monitor the level of cleanliness of the eggs coming into our hatchery egg rooms. Another method of traditional microbial plating is air plates. Obviously, these can be used to monitor how clean are our vaccine mixing rooms in our chick room after we've cleaned or before we've cleaned. And then another great method of microbial plating is the use of fluff counts. So this is basically using an air plate, but putting it in the hatchers while chicks are pipping and hatching to measure the amount of contamination found within the fluff that's collected on our Petri dishes. And this is a great way to, again, go back and double check the validity of our formaldehyde drop schedule or our hydrogen peroxide system schedule. There are a couple of downsides to traditional microbial plating, depending on if you process these samples in-house or send them off to a lab. It can sometimes take days or weeks to get your results back. Well, why is this a bad thing? If the point of traditional microbial plating or hatchery sanitation monitoring is to keep a check on how well our sanitation programs are working for us, what if we don't remember who cleaned incubator number 5 when we took this swab or this air plate? It's going to be difficult to explain to our sanitation teams and understand and maybe find who wasn't using the hot water that day or who wasn't using proper mechanical force. Another downside is that they can be expensive, so we might not sample every area of our hatchery on a regular basis. More traditional methods of monitoring: Bac-T swabs serve as a collection and transport medium to get bacteria to the lab for testing. And also boot swabs, which can be used in the hatchery as a great way to test for salmonella on various surfaces, clean chick boxes or your chick pool belts. One thing you can do instead of putting them on your boot and walking around is apply them to your hand and just rub the surface area where testing is desired. Kind of a new thing on the hatchery front is the use of ATP bioluminescent swabbing. We've seen these devices used for years in poultry processing plants, but it turns out they are applicable to our hatcheries. The way these work is by using ATP or adenosine triphosphate and luciferase, which is the chemical that allows lightning bugs to emit light. ATP binds with luciferase. The more ATP, the more luciferase, and the more light, and it puts out an RLU value, or relative light unit. So the higher the ATP, the more luciferase, the more light, the higher the RLU value. Now, these ATP bioluminescent swabs are great because they give us immediate results. Once you swab and place the swab in the device, it takes 15 seconds to give you your RLU value. And another good thing about the ATP bioluminescent swabs is that they can be adapted for use in any area of the hatchery. So these ATP machines, I believe, were designed to be used in very sterile environments, hospitals, nursing homes, processing plants once they have been cleaned. And so they come with guides for cleanliness. So for example, 0 to 20 RLUs is clean, 21 to 50 is okay, and anything above 50 we need to clean again. But we can adapt and we can set our own guidelines for any area of our hatchery and use these ATP swabs. One downside is the initial investment of the ATP bioluminometer that you see pictured here. There are several different kinds of swabs that can be used in the ATP bioluminometer, but one of the best ones for hatchery use is the UltraSnap ATP surface test. And these are designed to detect total ATP from any cell type on the surface that you swab. The reason these are great for hatchery use is because they're tolerant to temperature abuse. So we can take our bioluminometer out into the egg room with our pack of swabs, and we can make our way all the way through the hatchery, going in warm incubators, warm hatchers, or a very warm and humid tray wash or box wash room, and it's not going to affect the validity of the swabs. The shelf life for these is 15 months at refrigeration or no longer than 4 weeks at room temp, so plenty of time to use these swabs in our facilities before they expire. For the more visual folks in the room, I have just an example here of how the ATP machine actually works. So all cells contain adenosine triphosphate, which is the universal energy molecule for living cells. It just so happens that occasionally we see eggs like this in our hatcheries that have bacteria or microbes that contain adenosine triphosphate. So we take our swab and we swab the surface of the egg or the surface of insert here, whatever you want to test. And in the top of that swab is the luciferase enzyme. So once we collect our sample, we place the swab back in the tube and we break the snap cap, which allows the luciferase to come in contact with our sample. As luciferase binds with ATP, light will be emitted and we'll be given an RLU value or a measure of cleanliness in 15 seconds. So a great applicable use of these swabs in a poultry hatchery would be to test the cleanliness of eggshells. So we've always heard that, well, this breeder flock is older, so the eggs are dirtier. Is that really true? Maybe so. So this test was completed on 25 different eggs from 5 different age breeder flocks. And so you can see here that the 29-week-old flock is the blue flock, the blue line on the bottom, and the 33-week flock is the red line. They both had lower RLU values on eggshell swabs than the other 3 flocks, besides this one point here on egg number 4. You can see here that the eggs from the 57-week-old breeder flock had the higher RLU values across the board. And then in the middle here is the 36-week and the 43-week-old breeder flock. So just a very unique, applicable way to use the ATP swabs in the hatchery to monitor sanitation. And with that, I'll wrap it up really quickly. I hope that if you don't take anything away from this today, I hope that you can take these 2 key takeaways back to your facilities and implement them. The first one is know what you think you know. If you didn't document it, you didn't do it. Do we have the paperwork in place to keep track of our daily sanitation, cleaning, and disinfecting procedures? And then number 2 is just make it simple for your people. Don't just tell your folks to come in and, oh, today you've got to clean setters 1 through 5. explain why. Explain the importance of the 4 factors of clean and use the 4-legged stool model as an example for that. And with that, I appreciate everyone for listening, and I will take any questions.
Thank you. Very interesting discussion. You can unshare your screen now if you would like.
Okay.
Unless you just want to leave that up and promote.
That's fine.
Several questions are rolling in here. One of them kind of struck me. It says, what do you think about spraying a wet disinfectant like hydrogen peroxide after transfer? So between transfer, using a wet disinfectant, spraying it on the eggs. What is your thought on that at transfer?
As a given rule, we never want to spray a wet disinfectant on the egg because, as we know, eggs have pores, and so anything that we spray on the surface of the egg can come into the inside of the egg via those pores. So wouldn't recommend a wet spray without good proof that it's a good idea to do in our hatcheries.
I think the whole idea of spraying the eggs is for a long time. Don't get eggs wet. Don't get eggs wet. Well, it can be done if everything's done right. Like if your spray solution is warmer than the egg and it's not going to get inside the egg and all that stuff. But from experience, it's too easy to not do it right, and then it's worse.
That's right.
And it's worse. It ends up being bad. So, what is your opinion on using Veracid? It's considered a good, against bacteria and fungi and stuff, fumigating inside the machines.
Yeah, I think Veracid is a great disinfectant to use, whether you spray it or use it as a foaming agent. Veracid has been proved to work very well in poultry hatcheries.
So how about spraying it in the machines? Would you do that with or without eggs? I mean, I've gotten this question several times. Can we spray in the egg cabinet with eggs in it?
I think the key there would be to make sure that your spray is more of a fog than a spray. And if you do decide to spray liquid, make sure, or the fog, right? Make sure that you have a good check valve in place so that your liquid isn't dripping while it's not spraying, because again, we don't want to wet eggs. We don't want to wet chicks, but it never a bad idea to add a level of disinfect anywhere throughout the hatchery. Again, as long as we're not soaking the eggs, we're fogging the Veracid within our machines.
I mean, a lot of things, if it's done correctly, it's good. If it's not, then
That's right.
Okay, so somebody said in here they're already fumigating done on a daily basis. Would it be required to fumigate hatching eggs when they come to the hatchery? They're already doing it at the farm. Then they're coming to the hatchery.
That just depends on the hatchery, really. Several hatcheries are built with fumigation rooms in the egg room.
Mm-hmm.
Depending on what your company egg production looks like at any given time, you might be setting eggs right away when they come into the egg room. So I would say if you can get a good system down where you can fumigate in the hatchery, in the egg room, you have enough employees, enough labor. It's never a bad idea to add another level of disinfection.
Just kind of make sure you're using a good product, right product, and using it correctly. Because we have seen where people have used some of these and use them at too high of doses and different things, and that can always cause some problems.
Right. And do your research, call somebody that does it at their facility successfully. And the whole time that you want to implement a new process such as that, keep a check. Is it really making a difference? Are we monitoring the level of cleanliness before and after? And have we proven that it's worth our time and our money?
Right. I think over time, sometimes you see in hatcheries, not just hatcheries, everywhere, you keep adding all these things. We should do this, we should do this, we should do this. Sometimes we don't really know if we're getting anything out of it. And then pretty soon we got so many things we're doing, and it's like, okay, what's essential here? What's not? So I think that's probably where they're coming to. Do you think it is essential to fog throughout the hatchery for bacterial stuff like the rooms, halls? What's your thought on that?
I think so. Every hatchery, again, the common theme here is it's going to be different for every single hatchery. Just multi-stage versus single stage is going to put up lots of differences from hatchery to hatchery. But I think it's a great idea to create a schedule. Maybe if you don't have time or the people to fog every room, every hallway every week, rotate it throughout the month so that everything's getting cleaned once a month or every other month.
Okay. What's your thoughts on, or thoughts or suggestions on how to clean multi-stage incubators to reduce, the contamination, because they're in use most of the time. I mean, there's very little times they're out of eggs. What's your recommendations on that? What do y'all
The
do?
key, I would say the key there is keeping a great cleaning schedule, because we obviously have to plan ahead when we clean multi-stage setters, right? Because they have to be empty because we don't want the eggs to get wet. So cleaning those quarterly, 4 times a year would be the best. Lots of multi-stage machines are designed to have mats that go under the eggs, right? That catch the rots or the contamination, the busted egg yolk that falls. So cleaning those on a regular basis, but a deep clean of multi-stage incubators quarterly would be ideal.
So would you manage it to where you can get the eggs out to do a deep clean?
That's right.
So if you've got a time where you're not running full capacity, a lot of places, if they're running full capacity, they don't have a machine empty. But I'm like you, I that's always bothered me. I'm like you, so make it a plan, get it on a schedule to do
For
it.
sure.
Yeah, that could be a problem. What is your recommendations if you know the percentage of floor eggs is pretty high, but you've got to use them? How would you manage that from the farm to the hatchery? I mean, there's a number of ways to go with that. We tell people, oh, don't use these. Well, we have to. So they're going to use the eggs.
Well, many folks here probably know Dr. Gina Wilson. I've heard her say that if you bring in 1 floor egg in a flat of eggs, if that floor egg becomes contaminated, which it probably will, and it will probably explode, it's going to cover every single egg in that flat, which is going to reduce our hatch overall. So we might be setting more eggs, but we might be hatching less chicks because of the increased amount of contamination to a good hatching egg. So I'm with you. Use them if you have to, but Dr. Wilson would never say that. She would say nest clean eggs only. So.
Yeah, but I understand you tell people that. It's like, no, we got to have them. So, I mean,
Yeah.
if somebody has to have them, they have to set them. What are your recommendations? A lot of people just put them at the bottom. So we were talking with somebody the other day. They have enough of those. They have a whole machine that's all dirty eggs, the whole thing.
That would be ideal so that we're isolating the contamination. A whole machine separate would be the best if you have that many dirty eggs.
There's a problem if you have that many.
Yeah, there's an underlying issue there. But even if we set those dirty eggs on the bottom and they explode, some of that explosion is going to go up. But obviously it wouldn't be as bad as setting them on the bottom and all that contamination running down through the trays below.
Well, one thing you had said in your presentation that I kind of caught and grabbed a hold of because we've dealt with this is you've got chicks hatching, so there's maybe slightly on the green side, or they're just hatching their normal process, so their navels are a little bit open, not completely open. Maybe by the time they get to the farm, they're closed up, they look good. But if you have contamination in your machines and in your hatch baskets, they're hatching and crawling all over that hatch basket. And if you've got contamination there and that navel's not all the way healed,
exactly.
you've got a bad issue. And it might not show for 2 or 3 days in the farm is what we're seeing, but it's an issue.
That's right.
It's an issue. What about ozone? Have you done anything on ozone?
I haven't. I've read a couple of papers, but nothing really with ozone has stuck in a commercial setting.
Yeah. I mean, it's been around a while trying, but I haven't seen it used large scale at all. What about and you're cleaning your hatchery and stuff, is it necessary to do cleaning of the drains? Because of course, everything you're cleaning, you're flushing down a drain.
Right, that would be the last step. When we go from a top clean to a bottom clean, we definitely recommend cleaning out the drains for sure.
Okay, and just using that as you're doing your normal procedures. Like you said,
That's right.
put it on your schedule and do it.
It can be annoying to do that, right? Because little particles of eggshells will end up in the drains and then they're wet, and wet eggshells are a pain to clean up, but it is very necessary. Again, when we think about the umbrella example, those eggshells can just hide bacteria. So was our clean 100% successful if we don't finish the drill and clean out those drains? Maybe not.
Yeah. What's your opinion on using formalin in the hatchers, and what's an alternative use for that? I've got my own opinions on formalin, but what's yours?
I've not looked into using formalin, and I guess the question is as an alternative to formaldehyde, using formalin
alternative
liquid.
formaldehyde, yeah.
I haven't looked into that much. Obviously formaldehyde is a hot button topic. But when used correctly, is proven to work and be safer for our employees as opposed to if we're not using it correctly. So formalin might be the same if it's used correctly and appropriately in the hatchers.
Do you think if somebody's fogging, like a room fogging, can that spread asperspores?
That's a good question. I've not really thought about that much, but I would say that
I hadn't either. That's why I asked you.
Yeah, I would think it would kill the spores if you're foaming correctly or fogging, you said. So again,
I guess the thought is that if there's spores there in your room fogging, does that give an avenue to spread them more? I haven't seen any work on that.
No, I wouldn't think so, but
yeah. So I don't know. I just wanted your opinion. A lot of questions here on fogging and stuff, and we might address those later. You talked about water and sodium chloride in your webinar, and somebody wanted you to explain a little bit more about that.
Sure. So the PathoSans machine basically uses an electrochemical activation process with pure salt. So it's a very complicated system where water and NaCl move across a cell, and basically the cleaner is produced and the sanitizer is produced from that chemical reaction, and it's readily available for use. So it's a cool thing to look up and look into, safe for employees, and all you have to do is have water and pure salt.
Okay. And you had said in that discussion, like for multi-stage, you would like to see them clean quarterly, and of course, single stage, we know every time you get involved. Yeah. Okay. Yeah, that was one of the questions. Somebody wants some clarification on that. So the dry hydrogen peroxide, I know you've done some work with Dr. Jordan. Can you explain a little bit more about that, how it works, and what the potential benefits of that are?
Sure. So the dry hydrogen peroxide is a proprietary technology where the humidity in the air and the oxygen are brought together in the dry hydrogen peroxide machine. They're also moved across a photocatalytic membrane similar to the cell in the PathoSans, where the H2O and H2O2 molecule, the H2O from the water and the O2 from the air are split apart, and then they're joined back together in the form of hydrogen peroxide, which is H2O2. And so it's a completely gas phase of the molecule of hydrogen peroxide. So there's been several studies completed using dry hydrogen peroxide in hatcheries as an alternative to formaldehyde, because obviously the main setback from using liquid hydrogen peroxide is the corrosiveness to our hatch cabinets, which we don't want. So the thought is if we use dry hydrogen peroxide and there's not a liquid hydrogen peroxide running over our machines, it's a much lower concentration of peroxide, then maybe we won't see the corrosiveness of traditional liquid or fogged hydrogen peroxide. So it's a really cool thing. There are several papers published, and I would urge anyone interested to go check those out.
And so your concern with using regular hydrogen peroxide would be the corrosiveness on the machines. I mean, as a sanitizer, does it do well?
Works great. Works great as a sanitizer, but it can cause rust.
Yeah. If you're using formaldehyde in your hatcher, how would you go about measuring that to get, I mean, is it something people do? Is they measure the concentration of it, or they just have a dosage effect and they just do it and put it in, or do people actually measure it?
It depends on the hatchery. I know some hatcheries drip the formaldehyde into their hatch cabinets, you know, maybe 300 milliliters every 6 hours throughout the hatch process for a total of 6 drops of formaldehyde. Some people just fog it using a wick system. There are several different methods. You can, of course, test the concentration of formaldehyde in the air using, I forget what they're called. They're little safety test badges where you can test the concentration of formaldehyde. But the best thing to do there would be to truly monitor the level of kill would be, again, fluff counts, like I mentioned.
Okay.
So,
So that's the way they're going to manage. Without Clinifarm readily available, how would you recommend going about trying to control asper?
That's a great question.
Do you have a great answer for us?
I'll try. So I would start at the breeder farm level with asper, making sure that we're bringing clean eggs in as much as possible. I know we've discussed that. That can kind of be an issue based on your hatchery, but that's where it starts coming into our facilities. And so I would start there, clean eggs only, increasing our sanitation in the egg room after every single set, move-in buggies, cleaning the floors. But then the next step I would say would just be AON maintenance, filter checks, things like that.
Yeah.
I know that's a tough one.
Checking, like you said. I don't know how. One of the things I liked you said was door discipline, of keeping the door. I love that phrase, having door discipline. But also you talked about, you know, if it's not documented, it wasn't done. And one of our consultants had always said, inspect what you expect.
Exactly.
Document it, inspect it. Is it really being done? And then following through to make sure things are done the correct way. So, well, thank you very much. There's quite a few other questions here. We'll address those one-on-one after the fact. We'll send some of those to you, and we'll address some of them as well. So those of you that ask questions on here, we'll try and get a answer for you when we can. But very much appreciate your presentation. Some good information there. I think sanitation in the hatcheries is going to continually become more and more important as we deal with avian influenza and different things that obviously are not going away. So I think this is a very pertinent area. You're in a good field to continue this and help hatcheries all over the place. So thank you very much, Kylie, for your presentation today. Thank you, everybody else, for joining us on our webinar, and hope to see you next month at our webinar next month. So again, thank you very much, and hope you all have a very good day.